Hello, you're listening to Kopi Time, a podcast series on markets and economies from DBS Group Research. I am Taimur Beg, Chief Economist. Welcome to our 13th episode. In late February, when there were less than 4,500 cases of COVID-19 outside of China, Microsoft co-founder Bill Gates said, "The coronavirus is starting to behave like a once-in-a-century pathogen we have been worried about." Some accused Bill Gates at that point of fear-mongering, but he was, of course, spot on. Here at Kopi Time, we have spent many sessions discussing the economic and financial market outlook in the middle of this once-in-a-century pandemic. Today, we go to the heart of the matter, the epidemiology of COVID-19, and to discuss all the critical questions one might have, we have with us one of the finest minds in the field of zoonotic diseases, bat immunology, and pathogen discovery. Professor Wang Linfa is Director, Emerging Infectious Diseases program at Duke-NUS Medical School here in Singapore. He played a leading role in identifying bats as the natural host of the SARS virus way back in 2001, 2002, or 2003. His research then extended from bat-borne viruses to better understand virus-bat interaction and how bats coexist with a large number of viruses without developing clinical diseases. His recent research contributions include developing antibody-based serological tests to detect SARS-CoV-2 virus, responsible for the COVID-19 outbreak, and the early and successful culture of the virus from an infected patient sample. His team also is working together with local and international partners to develop new vaccines and therapeutics for COVID-19. Prof Wang is a member of multiple WHO committees on COVID-19. Prof Wang, welcome to Kopi Time.
Thank you for having me.
It's a great honor and pleasure for us. Let's begin with some very simple issues. I think for the general audience, let me also point out that SARS-CoV-2 is the virus and COVID-19 is the disease. But in terms of similarities, how similar is this virus to the SARS virus?
Yeah, so this is a good question because when it started, in January and late December and early January, I think the Chinese government went extreme lengths to say it's not SARS. Okay, so to a certain degree, it's still right? SARS stands for severe acute pneumonia respiratory syndrome, right? And we know now COVID-19, so this is the new name that WHO named as a disease, as you already pointed out in introduction. So 80 to 85% of the COVID-19 patients are not severe at all. So in that regard, it's not SARS. So the disease is called COVID-19. But it's called a SARS coronavirus 2, which is the agent which caused COVID-19. The reason that virologists decide to go that way is because we have a naming convention, right? So like every animals, including humans, in the taxonomy, you have these 4 different levels. You go to species, genus, and family and order. So at the species level, virus also have that. So we have one species we create called a SARS-related coronavirus. And then the SARS virus in 2002, 2003, and this COVID-19 virus in 2019 are actually very similar, okay? So for that reason, the virus is called a SARS coronavirus 2. So I hope that your audience can appreciate the complication there. The disease is different, and the virus is very similar.
That is an extremely useful distinction, and you actually helped me as well tremendously. Thank you for that.
Okay.
Okay, now I have to ask you more of an explosive question because this is being talked about in the US a lot. Are we sure about the origin of the virus, and does it really matter where it came from?
Yeah, so I think, as a scientist, I'm really saddened that such an important scientific question has been, politicized to the level, between the 2 major countries. To me, that as you mentioned, that actually I was the one discovered the origin of SARS. So 2003, I was seconded by WHO. So 17 years ago, we had a WHO team of international experts come together, really try to find the origin of SARS. So that was in July and August 2003. And the whole epidemic, of SARS was under control. And so we have scientists come from, France, USA, Japan, China, Australia, all work together, Philippines. And we discovered eventually, we have 3 nations, China, Australia, and USA, 5 Institute, we discovered the origin of SARS. But that, took a long time, and the really final conclusion came in 2012, 2013. So it's almost 10 years of effort. Wow. So I'm always, telling media people and scientists included, just be patient, because scientifically, it's not easy to identify the true origin of a totally unknown virus. So in this case, the best evidence we have is for this SARS coronavirus 2, the closest cousin, if you want to say that, is that a virus in bats, okay? But we know that bats all over the world carry similar viruses, okay? So this particular one was discovered in China, in Yunnan province, but that virus is 96% identical to the COVID-19 virus. And just give you some numbers, this SARS coronavirus 2, the genome has the genetic code we call nucleotide is 30,000. So even 4% difference means that there's 1,200 codes are different, okay? So I always say, there was a discussion about whether it's possible to engineer virus from the bat virus to the human virus. I say, in theory, you can. In practice, its chance is 0, because the probability of you get right, because as I said, it's a 1,200 difference, right? And then every code, there's a 4 possibility. You're mathematically much better than me. So it's a 1/4 to the power of 1,200. Most of your calculator will explode. You cannot get that number, basically. So the chance is almost 0 to have, even the smartest scientist to create a virus from the bat virus to the human virus. Your question of does it matter? It matters in long term, because we want to know where the virus comes from, especially the early transmission event, because we believe that maybe more than one animal is involved. One animal may be the natural reservoir, and the other animal is the intermediate host. By knowing this, hopefully you can prevent future outbreaks by similar virus. But as a lot of people discuss and agree, is that it's not the top priority right now. Over.
Yeah, absolutely. Yeah. So I want to take advantage of your expertise on this other issue, which is this extreme infectiousness of COVID-19. I've read that SARS-CoV-2 spike protein has the ability to cleave more readily than equivalent proteins in other coronaviruses. Can you explain that for the general audience?
Yeah, so in terms of infectivity, actually, there are many factors, where impact on the virus. So yes, you're doing a good job there, because this virus, like the SARS virus and like many other viruses, actually needs help. When the virus, make a contact with a host cell like human cells, they actually require something called a cleavage. So the protein on the surface of this coronavirus we call the spike protein. The spike protein needed to be cleaved in order for it to expose something that can penetrate the cell surface membrane and get into infected. But that's only one of the sort of determining factor. The other is called the receptor, right? So everybody, is talking about this ACE2 molecule, which is a protein on the cell surface. So the COVID-19 viruses are unique compared with the SARS coronavirus, for example. It has 2 features we think that it contributes to this more infectious nature. And one is what you already point out, that they have certain sequence on their spike protein, make the cleavage more efficient, so means more infectious. Secondly is that after this cleave, that the spike protein need to bind to this receptor ACE2, and then separate study found that binding affinity is also higher for COVID-19 than SARS. So maybe there's more than one factor contributed to this more infectious phenotype. Thank you.
I see. Then there is this issue of asymptomatic carriers. What do we know about this? Because, in the press, there's all this question about, there are stories of asymptomatic COVID-19 patients then being infectious. Do we know how long they're infectious yet? Or what is the state of the art knowledge on that issue?
Yeah, so this is obviously for the general public, it's very important. And for scientists, it's also very important. But I have to say that, this is another one of these questions, just like the origin of virus, is important, but it's difficult. The reason I say that is that when you have a virus infect a human or animal, what we call virus-host interaction, and we know that, let's just use human as a host, every individual, even if you have your twin brothers and sisters, they also have subtle difference in their body's defense system. Because of that, then you have the whole spectrum of presentations. You have, all the way from asymptomatic to death, right, for COVID-19. And we already know now more than 4 million people already got infected by this virus. So it's only natural that you will have a display of wide spectrum of these symptoms. So this is the first point I try to make. Asymptomatic infection is not unique for COVID-19. For any virus infection, if you have a large enough number, you will have a spectrum of severity from asymptomatic to severe to death, okay? So this is the first, I think it's very important to make. Secondly is that on the question of can asymptomatic sort of patients who are infected, can they be infectious and pass the virus to other people? The answer is definitely yes. But is that, occupies 1% or 10% of the asymptomatic patients? We don't know, because the exact number of that asymptomatic infection out of the total infection, we don't know. Then out of the asymptomatic infection, what the percentage will shed virus and become very infectious, we don't know. So I have to disappoint your listeners, because that the answer is that yes, it's possible, but exactly what's the percentage, I don't think scientifically we can say.
I see. Okay, that's cause for worry for sure.
Yeah,
because when we're talking about opening up from lockdown situations, if we don't have the capacity to test a lot of people, then of course these issues will come up. That asymptomatic carriers could be just in the general population. Prof, how are scientists testing for the virus, and how accurate are these tests? And of course, we keep on hearing about cheaper and faster tests in the pipeline. Where do we stand with that?
Yeah, so that's another good question, and no simple answer, right? So in terms of how you test an infection, you're saying how you test a virus. To me, is the more accurate description is how you test for infection, okay? So when you test for infection, you have two ways of doing it. One is you find the direct evidence of the virus in your body, okay? Now, 50 years ago, that's very difficult to do, okay? And now we have an invention called PCR, the polymerase chain reaction. So right. This is a molecular detection, actually zoom in right into the genetic material. So in this case, COVID-19 or the SARS-CoV-2, it's RNA virus. So you detect for the presence of RNA. So that's when in the early phase, in January, February, and March, when you think of people talking about tests, that's the only test, because people won't have evidence of the presence of the viral material. Okay, so the test is called a PCR, or the molecular test. So the test is very specific and very sensitive, really as good as you can get. Of course, because there are hundreds of companies marketing it, so nobody really have absolutely knowledge of to say test A is equal or much better or much worse than test B, unless you do all the test validation for hundreds of tests. And so overall, I would say that molecular test is very sensitive and very accurate. And then you have the second type of test. What we do is actually monitoring the immune response in your body. And you already mentioned in the introduction about serology. So serology is a kind of a discipline of science. Basically, translate into layman's term is to detect virus-specific antibodies or pathogen-specific antibody in your blood. Okay, so the advantage of the serology test is that humans or animals, we all have a defense system, right, in our body. So when we get infected by a virus like COVID-19, we try to clear them. And most time we win. So within like 10 days or 2 weeks, your body will already clear the virus. So the molecular test will no longer be used for because the genetic material is gone. With the antibody, once your immune system sees the virus and fight the virus, and they induce lots of defense molecules, one of them is called antibody in your blood. Antibody will last months, years, and sometimes forever, like measles virus. If you get infected by measles virus, the antibody stays in your body for the rest of your life. Okay, so these are just two different types of tests, molecular tests and antibody tests. Then we have, again, two groups of tests. One is more accurate, but require the sample to be sent to specific labs, like my lab or a hospital diagnostic lab. So this is what we call lab-based test. And what you're talking about, this fast, cheap, test, is what we call is a point of care test, POC, or rapid diagnostic test, RDT. So these are basically like a pregnancy test. How you have a cassette, and then you either using your sputum or your swabs from oral swab from your mouth, or you do a fingerprint, use a few drops of blood, and usually within half hour or 10 minutes you get the results. And by default, of course, these rapid tests are less accurate and than the lab tests. So it's pros and cons. I always say there's no such thing as a perfect test. You develop a test, utilize a test, and only fit for purpose. So if you want to have a rapid test that you can afford to be, 20% non-specific, then you go for the rapid. But if you say, I want 99% sure that if I have the virus or don't have the virus infection, then you go to a lab test. I hope that, give you a very, very comprehensive summary of the tests we are facing right now.
No, absolutely. And you began talking about antibody. Maybe we can spend a little bit of time talking about that, because I've been hearing some conflicting reports about the antibody not staying in people's system like the way they do for some other affliction. And therefore, it is possible for somebody who had COVID-19 to be reinfected. How true is this?
I think it's not very true, because for two reasons. One is it's really too early to make that conclusion, right? Again, just like when you say asymptomatic infection versus severity or death, when you have 4 million people infected, the antibody is just part of your host defense. So same as I said previously, that the human immune defense system varies so much from one individual to the next individual. So when you have 4 million people infected, you're going to get a really wide distribution. Yes, so we have seen patients recover from COVID-19, and yet they don't develop high levels of antibodies. And also, I'll take this opportunity to educate, your audience that there are two types of antibodies in your when you see a pathogen. Your body will have developed antibodies, but most of them we call is binding antibodies. The antibody can bind to the viral proteins. And there's a subset of these antibodies are more important. We call them neutralizing antibodies. So they actually can bind the virus, and then neutralize them means that they make them inactive or they could not infect the cell anymore. So usually we measure the neutralizing antibody as correlates of protection. So WHO is hesitating to say this because it's too early. So there are two questions what we need to address. First of all is that can we have a reliable test to test neutralizing antibodies from recovered COVID-19 patient? And then the second question is that can we be sure these neutralizing antibodies actually correlates with protection? Okay. So I'm working on the first part. We have just developed a novel test. We can measure neutralizing antibodies. But in terms of whether neutralizing antibodies is a good indicator of protective immunity, and that requires more time, really. So that's the first part of the answer to your question when people say, COVID-19 is less effective to produce neutralizing antibody, and so that, recovered patients can get reinfection. I think that's totally, speculation. And we don't have enough evidence to point that. Secondly, is that we always refer to SARS, right? SARS and COVID-19, as I said, they are basically close cousins, the same species of virus. We now have evidence to say from Singapore, we were quite badly hit by SARS coronavirus as well. So we have SARS survivors, this time in order to study COVID-19, we actually approach them to voluntarily donate blood so that we can check their blood to see 17 years later whether they still have neutralizing antibodies against the SARS coronavirus. Really indirectly to answer the question, because for COVID-19, if we wait for 17 years, that's a long time. But because the SARS is so close to COVID-19, so we did that study and we found that, they still have that antibody in their body. So that is indirectly to say that the conclusion to say COVID-19 may not have lasting antibodies is incorrect. But as a scientist, I would say that it's suggestive rather than conclusive, because the SARS data would suggest that actually COVID-19, the antibody will last in your body. But we will see, So we actually have, in Singapore, we have just, launched that study. We're going to do longitudinally that, from the recovered patients COVID-19, we're going to go back, ask them to, come back, and we will take blood samples, and we will do that study in 3 months, 6 months, 12 months, 18 months, and we will continue that. Thank you.
Absolutely fascinating. Okay, so related to the antibody issue, one more point or one final point on that issue, plasma from a cured patient. Is that being used to, treat existing patients? And where do we stand there?
Yes, and there's a specific terminology called a convalescent plasma therapy. So convalescent, it is a medical term, means that somebody has been infected and now is recovered, healthy. And the plasma, as you already know, is a part of the blood, which contains the antibody. And so this has been used for what we call passive immunotherapy, And so this has been used, obviously, first used by the Chinese because they had the patients first. So they start in February. And now, every other nation is looking into this. And there is a big company, it's a US-Australian joint venture, and I don't want to name names, but they already have a huge manufacturer, started, and they were asked for blood donations from recovered patients, and they're going to mass produce this convalescent plasma for therapy. And in Singapore, we're doing that as well. Okay, but again, one important aspect is that not all COVID-19 recovered patients will have good antibodies. So again, going back to my emphasis is to have a test which can measure the neutralizing antibody, so that at least give you an indication to say which plasma may have a high chance of being effective when used for passive immunotherapy. Thank you.
I see. Wow. We are reading about a wide range of antiviral drugs in development. My friend Dean Ho at N1 Institute at NUS published a paper last week where his AI has come up with a cocktail, I think large chunk of remdesivir and small chunks of two other FDA-approved drugs, and putting together, at least in his lab, it's showing potency against the virus. What's your sense of the most promising antivirals in the pipeline?
Yeah, so these two questions, the convalescent plasma, for example, is antiviral as well, right? So, right. So when you talk about therapies, so this is the antiviral is one area, so that you can have at least 3 different types of antivirus. So what you're talking about is what we call small molecule drugs, like, remdesivir, even chloroquine, Trump was really promoting chloroquine, right? So there are many of these small molecule drugs, either de novely developed or mostly what we call is repurposed, right? Chloroquine was developed for malaria, and remdesivir was initially developed for Ebola virus. And certainly that I think the front runner is, I think, according to US NIH trial, is the remdesivir. But there's some controversy there because there's 3 trials. The US trial says very effective, and the Chinese trial say not. And then you also introduce the concept of cocktail, right? So cocktail, obviously, that it's going to be more expensive and it's hard to control, but just by, sixth sense, it's going to work better because there are synergies there. When you have a 1 weak drug, used alone is weak. And then so you have drug A is weak, drug B is weak, drug C is weak. But when you put them together, they have synergistic effect, right? They can become, 3 weak, become very strong. So cocktail certainly is attractive. And then there's the third class of molecule is still antibody, but not from patient serum. So this what we call is recombinant antibodies. So these are actually in vitro engineered protein antibodies that mimics the antibody in your plasma. So quality-wise, it's much more cleaner because it does not have the blood composition in it. And secondly, you can mass produce it, and then batch variation is minimal. So you basically have these three large groups, okay? The first is what you were talking about, we call small molecules antiviral drugs.
Right.
The second is the recombinant engineered antibodies, single antibodies, but purified. The third is multiple antibodies, we call polyclonals, because there's many antibodies in your plasma. So all three, I think, currently the plasma and the small molecule drug is already in clinical use. Very soon, I think, in the next 2 to 3 months, there will be clinical trials of these recombinant monoclonal antibodies. And then there are also people trying these cocktails. You have been talking about cocktails within the same class, the small molecules, right? There have been discussion about cocktails across 3 classes. How about you use remdesivir plus convalescent plasma, okay? And so this is still an open field for trial, and certainly there are clinical trials ongoing, trying to test all different combinations. Thank you.
So parallel to all this heroic effort in terms of coming up with antivirals, we also need sooner or later a vaccine. Is it going to be really a couple of years, or is there some light at the end of the tunnel that maybe early 2021 or maybe even late 2020, we have some encouraging development?
Yeah, so again, I think that's is the most important question in terms of a COVID-19 pandemic, because what we have discovered or discussed, whether it's small molecule or recombinant antibody or convalescent plasma, these are all therapy, right? You're already infected and you may already be very sick, and before you see a doctor, and then the doctor tried to save your life by treatment. Much better is what we call active immunization, right? Active immunization means that you vaccinate, okay? So vaccine, everybody agrees, from NIH to most of the scientists in the field, is that in order to control the COVID-19 pandemic, lockdown is temporary, long-term solution is vaccine, okay? So as you can imagine, there are hundreds of groups doing vaccine studies, and then there are maybe 80 companies all claim have a vaccine candidates, and there are at least 10 to 20 are seriously planning what we call clinical trials. And currently, there are a few companies already in or finished. So for clinical trials, we have 3 phases. We call phase 1, phase 2, and a phase 3 trial.
Right.
Okay. Phase 1 is mainly for safety, and the phase 2 is really looking for the all the indicators to the vaccines working. And the phase 3 is really that you vaccinate the population, there's a high chance of infection, and then you see the protection. Okay, so currently that there are, as I said, multiple, maybe 3 or 4 in phase 1, and 2 of them finished phase 1. And then there's another company that another vaccine candidate is already in phase 2. So if everything goes well, by September, we will have the front runners have finished maybe all 3 phases of trial. And then, depends on, I always say, deployment of a vaccine during pandemic is a complicated decision-making process. Because this is both a scientific decision, public health decision, also a political decision, right? Just like we're facing with lockdown versus like in Singapore we have a circuit breaker, in other countries we have lockdown. So when do you lift the lockdown. Correct. It's a very, very complicated process and a decision making. Same for vaccine, because most of the human vaccines takes years and years to develop. So what we call this now is in the category of emergency vaccines. So we need to have different rules to apply for this. So that means that the person make the decision have to have some political, authority and responsibility, because it means that if we want to use the emergency vaccine, for example, in September or December this year, the risk will be higher than if you do all the tests and trials and then by end of 2022, for example. But there is a pros and a cons, right? So you have to, make this kind of decision. So I would say that September to December this year, there is a possibility of emergency vaccine approved for use, but it will not be a wild kind of international, because again, this is a national decision, because, for example, one country decide to use it, other country may decide to say, no, it's not worth the risk, and we just do more social distancing and, other containment strategies. So again, it's a complicated process, but the fastest, I think, decision can happen between September to December this year.
Prof Wang, what do you mean by risks?
Risks is that, like any vaccines, right? Because the vaccine is you inject a foreign material into your body. Just like, as I say, if a virus infects a human, that if you have 1 million people, you have a different presentations, right? So you have a asymptomatic to severe or even death. Vaccination is the same. Basically, we are trying to modify your immune defense, and then there are vaccine-induced side effects, right, in humans. Okay, and then for the coronavirus vaccine, there's even talking about, just like a dengue, I don't know how familiar you're with dengue, after 50 years of research into a dengue vaccine, we still don't have one. The reason is that dengue has 4 different serotypes. So if you have dengue 1, if vaccinated against dengue 1, you're protected against dengue 1, but you're not protected against dengue 2, 3, 4. And there's even talk about this enhancement called antibody-dependent enhancement, ADE. So there are discussions about whether ADE can also play a role for coronavirus. So risk comes in 2 different forms. One is that when you receive a vaccine, somebody may have a side effect, okay? And secondly is that a vaccine that you induce antibodies, if the antibodies is not neutralizing, not functionally neutralizing, you may predispose the person to future infections that make the disease even more severe. So these are all scientific discussions that we don't have evidence for or against that. But obviously for vaccine development, you have to take that into consideration. So these are 2 different types of risk. One is short-term risk. After you receive a vaccine, you may have side effect. The other is long-term risk. Thank you. Yeah,
personally, that makes me a bit pessimistic about a worldwide rollout of a vaccine in 2020.
Yeah, I think that 2020 is optimistic. I think maybe a certain nation may want to do that. But for internationally, I think, if we can have one by middle of 2021 for widespread use, international use. The other thing is really whoever developed the first vaccine, there's a huge pressure, right? You need a few billion doses if you want to vaccinate the whole world, right? So that's not easy to produce, yeah.
Absolutely. I think China would be number 1 in that category in terms of capabilities. Prof Wang, the one big fear is that this thing, just like the 1919 pandemic, influenza pandemic that happened, is that the coronavirus returns in waves. Any insight on that issue? Could this come back in waves, or it doesn't have to be that way?
Oh, I think that, without any doubt, like even in Wuhan, they have such a strict shutdown and they have 0 cases for, weeks, right? And then this week they had cases now, right? And same, for Melbourne, Australia, that they will have 0 cases and then suddenly there's a cluster. So these are almost like the spikes of, files, that if you have a very rapid diagnostic and the tracing, then you can distinguish the file in the spike, sort of a spark stage. But if you let it go for too long, then you may have a wave, right? So theoretically, a coronavirus second wave, third wave is still possible, certainly possible. And it looks like, the winter season will be the one that people most worried about. That's why, everybody is watching, WHO is watching for Australia and New Zealand, the two nations in the southern hemisphere. So we will see, right? Because both nations were so successful in control the first wave, but economically, they could not sustain this kind of lockdown. So I think schools are going to return and the people are going to, go back to work. And happen to be the winter is coming, in New Zealand and Australia. So I think that WHO and the whole world is going to watch these two countries and then to learn from their lessons and then to prepare for the countries in the northern hemisphere. Thank you.
No, this is a very big source of concern for policymakers. No question about that. Prof. Wang, I want to end with a broad, big picture question. You've been at the forefront of dealing with pandemics all the way back to SARS to today. What have we been doing wrong in terms of public health systems, in terms of public policy, that every 5-10 years something like this happens and we're caught off guard and then we scramble? What would you like to see governments around the world do going forward so that next time, inevitably, when another pandemic comes, we're better prepared?
Yeah, so the first thing, I think is that at the national level, I have been promoting this concept of wartime and peacetime, okay? In our defense, use Singapore as an example. For example, the military service training is in our DNA, right? All males, have to do military service. And let's face it, we are in such a peacetime, yet we are prepared for war, okay?
Right.
And we have a huge budget. We have a huge budget doing that. I sometimes joking about if we do the exact same for emerging infectious disease, just like a pandemic like that, if every nation treat this pandemic threat as military threat, and we use the exact same mechanism of funding and the training during peacetime, even with 10% of military budget, we can do much, much better. Okay, so that's the first point. At the national level, I always say, for this like outbreak, of course, scientists like myself, we strive, try to improve the detection, improve prediction. We can do, this, but at the end of the day, it's really at a high level, political level, funding, infrastructure, exercise, militarily, you have joined, a military exercise, all over the world, right? But for us, we don't exercise for pandemic preparedness enough as military, and we have to do that. And that, I have been promoting this for the last 20 years. We just have to treat the pandemic threat as a military threat. This is the first part. The second part is more difficult. Military conflict usually is bilateral. So, one country against other, or maybe 3 or 4 countries. But for pandemic like COVID-19, unfortunately, it's every country. Right now, we have more than 200 countries already infected, right? So the cliche says virus recognizes no borders. So this adds to the second complication is that international collaboration. And WHO remains to be the best organization. Yet, we already know that politically now it's how difficult it is, for everybody to agree to coordinate by WHO. So I think that the challenge is huge, but to me, the direction is clear. National level, I think that during peacetime, you have to prepare for the war. Otherwise, you will never win the war. And then secondly, internationally, we have to recognize, this is no like a military conflict. pandemic is going to affect everybody. And unless we work together, we will never win. Yeah, thank you.
Absolutely. Couldn't have put it better myself. Professor Wang, you may have heard, and maybe you're seeing it from your apartment too, that as we speak, there is a torrential rain and thunderstorm all over Singapore. Yeah. We hope that because of the diligence of people like you, that this storm will literally pass, and the COVID-19 storm will pass too, and we'll see some sunlight at some point in 2020, but definitely in 2021. Yeah. Thank you so much for your insights and perspectives.
Thank you for having me.
No, it's a great pleasure. You've been listening to Kopi Time. Thanks for joining us. You can find all our publications in multimedia by Googling DBS Group Research.